Can you now hear me? Is
Yes,
anything
we can
there?
hear you.
Okay, great. Uh, okay, let's begin. I don't think I need to say anything other than what we're going to do. Shut that page down. So today we're going to be talking about Lab 5, and you'll do Lab 5, Streaking for Isolation and Culture Media. You'll note that we don't have anything scheduled for Thursday, other than you do need to read the unknown project instructions file in Canvas. And I'll show you, well, I shall probably show you tonight because I don't think a lot of you will be here on Thursday. Let's see if I can get that. So let's go down to week 5 lab. And I better have part of week 5 lab shut down. I got to turn that on. There's lecture, there's lab. Yep, I need to turn on the unknown tests. They were so big, I thought you'd be scared if you saw all that, but you won't be doing all those tests. Those are just possible tests you could do. And you only need to do the ones to help you identify your unknown bacteria. All right. So the unknown project instruction files is on Canvas under week 5 lab. Oops, wrong one. And that would be the unknown instructions. So please read that and I will talk about that. Oh, good. The lecture lesson just finished. That's pretty good. That's less than 15 minutes. Hopefully I got everything there. Anyways, read that. That's the instruction for the unknown project. We'll talk about that project next week. And you could read that unknown instructions maybe on Thursday. I will show up to answer questions next Thursday in the lab. So I'll be there from 6:30 to 6:45, and I will be longer if people have questions. But if there's no questions at 6:45, I will log off. I guess that's here, sorry. Any questions about any of that? The unknown project is a project you will be starting week 6. I'll discuss it on Tuesday, and then we will begin the project and you will be working on it nearly to the end of the term. And it will take you that long to get it done. Where's my mouse? It's going to be turned in Saturday, March 4th. It's a 75-point project. It's all going to be done online, but you're going to be given a bacteria number, which is going to represent a bacteria species. And then you're going to try and identify the bacteria species by running specific tests. And I think I showed you some of them. To identify the bacteria species. These are some of the tests you could run. It's up to you which tests you want to run. It's all going to be done online. So I'll give you the bacteria number and you'll say, number 11, I think that might be whatever. And you will be given your first 3 tests. I'll talk more about that later. And then after that, you will figure out what the possible species could be and decide what tests you want to do, and you will request a test. Like you could request, I want the catalase test, and you will request that online, and then I will give you the test online, and it will all be done other than my first email. It will all be done in the unknown notes page, which I'll talk about next week. Any questions? If not, let's get on to the lab for today. And shut that down. Our lab for today is lab 5, cultivation of bacteria, culture media, inoculation, and isolation. I don't know if you can see that, but my glasses are a little dirty. There is some reading you should do before you do this lab. Page 157 to 162, talking about culture media. We'll talk about that today. And page 162 to 163, obtaining pure cultures. There is some video clips as supplemental material. There's no virtual lab today. Primary objectives. Upon successful completion of the lab, students should be able to understand the importance of proper specimen collection and transport to the laboratory, to be able to explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between the various types of culture media. 4, be able to explain the purpose of streak plate culture technique, aka streaking for colony isolation. 5, be able to define the following terms: what's selective media, differential media, and reducing media. Can you guys hear me? I got a note saying the internet connection is unstable.
It's a little patchy.
Okay. Like I said, just let me know and I'll repeat something. The ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, drying out, or exposure to oxygen, anaerobes, and require special transport medium, as well as procedures. Once the specimen is received by a lab for microbial culture, microbiologists employ the 5 I steps to process it. The 5 I steps are inoculation, isolation, incubation, inspection, and lastly, identification. Inoculation is just placing the specimen on an artificial culture medium to initiate growth. So you streak out your bacteria on a plate and get it to grow. Isolation, you spatially separate out the microbial growth into distinct colonies. You need distinct colonies to work with pure cultures. Incubation, placing the culture medium in an environment under conditions to facilitate its growth, meaning we'll put them in an incubator to grow the culture. 5, you inspect, examining the culture for the presence of microbial growth and certain colony characteristics. And then lastly, 5, identification, performing specific tests to identify any pathogenic microbes that may be isolated. There are different types of culture media. You're already familiar with one of them, general purpose media, including NA or nutrient agar, or NAYE, N-A-Y-E, nutrient agar with yeast extract added, and then TSA or tryptic soy agar. Those are general purpose media that grow many different organisms cheaply, but they do not grow everything. There are some fastidious microbes that will not grow on a general purpose media. And to promote their growth, you have to use enriched general purpose media. For example, you can add 5% sheep's blood to tryptic soy agar to get a growth medium to help organisms grow. And that actually is a blood agar plate, or BAP. Another enriched general purpose media is chocolate agar plates or cap. All right, so there are general purpose media and then enriched general purpose media, which all of these are non-selective. There's also selective media that contains chemicals that inhibit or halt the growth of some organisms. You should keep in mind when you're growing things on a selective media that the selective media isn't necessarily perfectly selective, meaning you may get some growth on the media, even though the bacteria are selected against. The selection won't be 100%, and so the bacteria will slowly grow on the media. And then second, even if the organisms are supposed to grow on the selective media, they may form smaller colonies or may grow more slowly than they would on other media, like general purpose media. All right, so there's selective media and then there's differential media. Differential media is a media that contains chemicals that produces visible differences among different groups of bacteria or organisms when they're grown on that media. The differences are unrelated to how well the organisms will grow on the media. Differential culture usually allows dissimilar bacteria to be visually distinguished from each other. Like one species may be one color and another species will be another color on a differential media. That would be one difference. Any questions about any of that? Lastly, there's reducing media, and this media is to promote the growth of anaerobic bacteria. Anaerobic bacteria require low or no oxygen conditions in order to grow. All right, so those are the different types of media. You should know that the culture media may be classified into more than one category. For example, McConkey's agar is both selective and differential. And here we're looking at McConkey's agar. It's selective in that it allows gram negatives to grow on it while it inhibits gram positives. So if there's growth on this plate, it's most likely gram negative organisms. And it's differential. On one side, the species is pink. On the other side, the different species is white. The pinkish bacteria is one that can ferment the sugar lactose, and that will change the pH and make the color pink. And the non-lactose fermenter will be not pink. In this one, it's its normal color, which is off-white. So this is differential and selective. Selective, and then only gram negatives will grow on it. Differential on the basis of lactose fermentation, positive, and this one cannot ferment lactose. Another one is blood agar, which is both enriched, an enriched media, and a differential media. It's enriched in that organisms which are fastidious cannot grow on general purpose media. Some of them can grow on blood agar. For example, Streptococcus pyogenes, which strep throat, can grow on blood agar, but cannot grow on a general purpose media. Streptococcus pyogenes needs something in the blood in order to grow. So that's why it is enriched, and enriched media. It's also differential in that you can differentiate between different hemolytic activities. There's beta hemolysis, which is complete hemolysis of the blood. Hemolysis is lysing the blood cells, and the white beta is the bacteria that was streaked on the plate, and then the clear region around it is beta hemolysis or complete hemolysis. And that's why it's not red, it's actually clear. You can actually hold this up to the light and see through it in the region of the complete beta hemolysis. Any question about that? There's also a partial hemolysis, and that's called alpha hemolysis. Let me blow this up a little. You have partial hemolysis of the red blood cells, and you can see that the white or off-white color is the bacteria, and you'll notice that the red from the blood does not come all the way up to the bacteria. We have a ring of darkening around the alpha. And inside the alpha, the entire region is dark, and that's from the partial hemolysis of blood, making it look dark. It may occasionally look green under partial hemolysis, and I'm not sure why sometimes it's just dark and other times it's green. It might be just the way you're looking at it. It might have something to do with the blood being lysed and how much is lysed. I don't know. Anyways, that's alpha hemolysis, partial hemolysis. And then we have gamma hemolysis, which is no hemolysis. And you can see that this is a gamma, the lowercase gamma in the Greek alphabet, and it's a white color, and the red comes right up to the bacteria. So no hemolysis or gamma hemolysis. And that's differential hemolytic activity on a blood agar plate. Any questions about any of that? Oops, I didn't mean to move that. Oh, well, make sure I don't save this. Let me shrink that down. All right, this table is summarizing the different types of media, general purpose media, in which case there are 3, TSA, NA, and NA, promotes the growth of most organisms, but not fastidious organisms. To grow on a general purpose, they have to be non-fastidious. And then we have enriched media, which the first one is both differential and enriched, and that's blood agar plates. It's tryptic soy agar supplemented with 5% sheep's red blood cells. And that promotes the growth of fastidious organisms, particularly members of Streptococcus, like Streptococcus pyogenes. And you can also look at the differences in the hemolytic activity on the blood agar plate. Another enriched media is CAP, chocolate agar. It's the same as blood agar, except the blood is added when the agar is fresh out of the autoclave, meaning the agar is still really hot. And that lyses the blood, so it no longer looks red, it looks brown. And that reminded someone of chocolate, and that's why it's called chocolate agar. So it's an enriched media. It's sometimes used to grow Haemophilus or Neisseria gonorrhoeae, which are fastidious organisms that do not grow on blood agar because these organisms cannot lyse the blood to get the nutrients. So they need the blood to be lysed already on chocolate agar before those organisms will grow. Oops, went too far. Then our next one is selective and differential media, and that includes McConkey's, also called MAC, eosine methylene blue, also called EMB, hectone agar called HE agar, and they're both selective and differential. They're selective in that they are used to isolate gram-negative, mostly gram-negative bacilli. They select against gram-positives. They're differential on their basis of their ability to ferment lactose. They'll be a different color if they can ferment the sugar lactose than if they cannot ferment the sugar lactose. And the color will be dependent on what plate we're talking about. With HE, the color is salmon colored if they can ferment the sugar lactose. With McConkeys, they'll be pink if they can ferment the sugar lactose. And I think on E and B, it's either a metallic green or a dark color if they can ferment the sugar lactose. And the color is generally the regular color, off-white, if they do not ferment the sugar lactose. But if you use a bacteria that has a pigment, like one that's red pigmented, the one that if it does not ferment the sugar lactose, it will be its reddish color on whichever plate you grow it on. All right, then we have another one that's only selective, and BEA, or phenyl ethyl alcohol. It's used to isolate gram-positive bacteria, meaning it selects against gram-negative bacteria. So this one's only selective. And then we have mannitol salt agar, which is both selective and differential. It is selective in that it has 7% sodium chloride in it. Mannitol salt agar has sodium chloride in it. And if the organism cannot grow in 7% sodium chloride, then it will not grow on mannitol salt agar. And if you don't remember, most bacteria will not grow above about 2% sodium chloride. So a lot of bacteria do not grow on mannitol salt agar. Only if they're salt tolerant would they grow on mannitol salt agar. So that's why it's selective. It's also differential on its ability to ferment the sugar mannitol. And the plate turns yellow if the bacteria can ferment the sugar mannitol. If the bacteria cannot ferment the sugar mannitol, it's not yellow, and it will be whatever color the bacteria is. So I guess you have to be careful if your bacteria is a yellow color and then you're growing it on this mannitol salt agar plate. This allows you to distinguish Staphylococcus aureus from other Staphylococcus species. Staphylococcus species can grow on mannitol salt agar, but only Staphylococcus aureus is yellow on mannitol salt agar because it can ferment the sugar mannitol. The other members of the Staphylococcus species family do not ferment the sugar mannitol. All right, here's some microbes we've grown. For isolating bacteria, you need to do that and get a peer culture. Like when you're studying your unknown, if it were to be a mixed culture, you'd have to streak it out and make sure you get a pure culture before you start studying it. Because if it has 2 or more species, if the test is positive for a result, you don't know which species made the test positive. So you have to work with generally a pure culture. Louis Pasteur initially developed this idea, and he developed a way of isolating bacteria to get a pure culture, but it wasn't very efficient. He used limiting dilution, and he would make several different concentrations of the bacteria, meaning several different dilutions, and then have 10 tubes of each dilution. And if he had one dilution where only one tube had growth in it, he would use that as his supposedly pure culture, because 9 tubes did not have any growth. And so the one tube that had growth meant that only a few or one cell was put into that tube to start it. And if you get growth from one cell, you know you have a pure culture. Like I said, that wasn't a very easy way to get a pure culture. And Robert Koch developed a much easier way to get a pure culture. And what he did was he simply streaked the bacteria on a solid media, meaning a plate of agar, and got isolated colonies. You then take the cells from that isolated colony and you streak it again for isolated colonies on a second agar plate. And if you get another isolated colony, you can use the cells from that isolated colony that went through 2 isolated colonies, meaning it was purified 2 times, and then you're greater than 95% chance that you have a pure culture. If you only go through 1 colony streaking, your chance of getting a pure culture is only about 70%. And that's just not good enough in microbiology. Any question about that? So Robert Koch developed the streak plate method, which was a very easy way to get a pure culture. And here we're using colony streaking on a plate. And as you can see, we streaked out the bacteria first in sector 1, and then in sector 2, then in sector 3, and then lastly in sector 4. And as you can see, the point of doing the different sectors is that someplace along the line, you'll be streaking out isolated cells, which will be streaked out far enough that they'll give rise to isolated colonies. And in this case, it was sector 4 where that happened, but you don't really care where it happens. It could be in sector 4, in sector 3, in sector 2, or maybe even in sector 1. Wherever you get isolated colonies, that's what you want when you're streaking for colony isolation. I suppose I should mention how you do that. So let me explain. So this we're going to pretend is a loop. Maybe I should go like that. And this will be my plate. What you do is you pick up your bacteria from your stock culture, and then I should use a rounder thing. Maybe we can do this. And then you streak your bacteria in sector 1. Okay, so you sterilize the loop, pick up the bacteria, and then streak it in sector 1. Wherever you first started, there will likely be a lot of bacteria, which was probably right here. And don't ask me why. They should have started here and went that way. But it looks like there's more bacteria here than there. And then you take your loop and you put it in the bacto incinerator to kill all the cells, the bacteria that are on it. And then you come over to sector 2 and you initially start someplace outside where the bacteria are. And then you go into sector 1 3 times. So if sector 1 is over here, you go in 3 times to pick up cells from sector 1. And then you streak it out in sector 2. So this region will have a lot of bacteria because we're getting the cells from sector 1. And I'm not sure why they went so many times in sector 1. You should only go 3 times, and they went 1, 2, 3, 4, 5 times. And another thing you should do is you shouldn't go all the way back in sector 1. You should be, because you're very heavy in concentration in this region. And as you're coming this way, it's lighter in concentration. So when you do sector 2, you should only streak where the bacteria are lighter in sector 1, not go where the bacteria are very heavy. And then streak out sector 2. And I guess that's there. And then you do sector 3. This is better because when they did sector 3, you take the loop from sector 2 and you put it on the background incinerator to kill everything. And then you bring the loop down to sector 3, and this actually shows it here. And you put it where there's no cells from sector 2, and then you go in sector 2. Now you shouldn't go all the way back to sector 2. You should be in sector 2 where the cells are lighter in this region. Don't go back where they're really heavy in this region. And then come in here and go through this 3 times. And they did it a little more than 3 times. But like I said, you shouldn't go all the way back. You should only be where the cells are a little lighter in sector 2. And then streak out sector 3. Okay, so I sort of showed it on the map there. Streak out sector 3. And then take your loop, put it in the background incinerator, kill off the cells that are on it, and then put it in sector 4 and bring it back to sector 3 3 times. 1, 2, 3, 4, 5. For some reason, they're doing it 5 times instead of 3. You should only do it 3 times. Pick up cells 3 times, and don't go, well, this is pretty good. They went mostly in this region where sector 3 is a little lighter. They didn't go way back here where it's really heavy. And pick up your cells from sector 3, and then streak out, after you go in 3 times, streak out sector 4. And that way you'll have sector 4. And as you saw, we got isolated colonies in sector 4. Any questions about that? Remember, this is only one colony isolation event. To make sure you have a pure colony, you should pick up cells from one colony here and then streak it out on another plate for isolated colonies. And after you've got cells that went through 2 colony isolation events, you can say with greater than 95% chance that that will be a pure culture. All right, so incubation is the next step. So while the culture media containing the nutrients is ready for growth, microbiologists provide an appropriate environment for microbes to grow. In our incubators, we only grow them at a warmer temperature and more humidity, but you can have a fancy incubator that is regulating how much oxygen is going in and how much CO2 is available. And this is a very fancy incubator where you put this whole container inside the incubator and you are controlling how much oxygen is in here. This has a chemical which removes oxygen, and so we can grow anaerobes in this anaerobic jar inside the incubator. And then you grow them for a certain amount of time, at least overnight. Usually in the lab, we grew them for 2 days. So we would streak them out on Tuesday and then look at them on Thursday. Or if you did Monday, and then Wednesday. You then inspect the colonies for obvious visible growth. If you have no growth, then there's something wrong with your clinical specimen, or there is something wrong in your handling of it. You can also describe the bacteria colonies by different things, like their different size, their different shape, their different margin, and the elevation. And you can refer to Lab 1 for a review of the appropriate terms to describe a colony. That will not be enough, though, to identify the bacteria species. So you then have to go to step 5, identification, where you run specific tests to get the results from that test to determine what the bacteria species is. And you will do that when you're doing your unknown project. All right, know the terms of this lab. And then go ahead and do the laboratory exercises. Let me go to the worksheet now. Oh, not quite. So there's a little video to watch on the inoculation of an agar plate using street plate culture technique. Only watch to 4 minutes and 15 seconds. After that, you don't need to watch it. You should note that this person is only doing 3 sectors, not 4, and you can do either 3 or 4. And then watch a little video explaining beta hemolysis, alpha hemolysis, and gamma hemolysis, which happen on blood agar plates. And then watch a little video on McConkey's agar. You only need to go to 3 minutes and 50 seconds. And then we have a little video on mannitol salt agar. And unfortunately, we do not have a virtual lab because the one we had requires Adobe Flash. And for some reason, the maker of Adobe Flash decided to discontinue it. And we never found another virtual lab for you guys to use. All right, so then answer the questions in the worksheet. They may be different in the lab module, so use the worksheet. Describe the natural circumstance where Staphylococcus species of bacteria might benefit from salt tolerance. So don't tell me something like, when you grow Staphylococcus on mannitol salt agar, it will grow because it's salt tolerant, okay? Don't do that, because that's not a natural circumstance. That's when you're growing it in a lab on a specific plate of media. So think of a natural circumstance where Staphylococcus is exposed to salt. And if you don't know, Staphylococcus will tolerate salt. And then question 2, how might hemolysis benefit a hemolytic bacteria organism? So we have a bacteria that can lyse the blood cells, hemolytic activity. And what benefit does that give the bacteria? And 3, imagine you have a culture containing a single bacterial species. The culture may be one of several possible species. Describe where you use mannitol salt agar, blood agar, and EMB plates to help identify the unknown bacterial species. That is, how would you use these plates to help you identify the species? And I'll warn you, to do this, you need to talk about 2 properties from each that you can use to help identify the species. Okay. Blood agar does have 2 properties, and that is you can get a fastidious organism to grow on the blood agar. And the second thing is you can look for the hemolytic pattern of the bacteria growing on the blood agar. So that's 2 things there, but name 2 things with mannitol salt agar and 2 things with EMB. 4, what is the main goal for streaking for isolation? Meaning when you're streaking out for colony isolation, what's the main goal you're trying to do? 5, what happens to the growth on the plate if you forget to sterilize the loop in between the sectors when you're streaking for isolation? You need to be specific, discussing each sector so that I understand your answer. Let me go back to the colony streaking picture. So this is showing you a plate where we sterilized, actually just put the loop in the bacto incinerator between each sector. And the question is, what will it look like if you don't do that? And you just take from sector 1, take your bacteria out of the stock culture and spread it in sector 1, and then you take this same loop and you start spreading sector 2, and then you take the same loop and you start spreading sector 3, and you take the same loop and you start spreading sector 4. Okay? So tell me what it'll look like. Oops, wrong one. And then in step 7, suppose you successfully streak a plate and obtain ice. I'm missing something. Number 6. Oh, here we go. Number 6, what happens to the growth on the plate if you transfer from the stock culture to inoculate each of the sectors? So in the picture here, to get sector 1, we put the loop in the stock culture and took the bacteria onto the plate and streaked out sector 1. But we did not go into the stock culture to streak out sector 2. The cells from sector 2 came from sector 1. And then the cells from sector 3 came from sector 2. What will it look like if for each sector you go to the stock culture and then you streak out the stock? So your discussion of 5 and 6 should be different. If it's not different, I'll ask you, how is number 6 different from the number 5? All right. Okay, suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species, that is from a mixed culture. How can you use this plate of isolated colonies to create a new pure culture of only one of the bacteria? Be specific in your answer. We used to have a picture of that. I don't think I have it here, though. Yeah, it's probably in a future lab. I got that picture. Eight, in the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. Denote on each medium, denote whether each medium is non-selective, selective, or differential by placing an X in the appropriate column. You may use the table of media in the lab module to answer these questions. So nutrient agar, if you think it's selective, put an X here. If you think it's differential, put an X here. If you think it's non-selective, put an X there. And do that for each of these different medias. You can look in the lab module that tells you. And then 9, we have 3 different plates and asking which agar plate was inoculated using a streak plate technique. So say A, B, or C. And that's it. Go ahead and start working on the lab. I will be here until 8 o'clock or the last student logs off. But once the last student is logging off, I'm going to log off too. Any questions?
I
Okay.
had a question about clarifying with number 6.
Uh-huh.
So I was curious for number 6, if just to clarify, is the question asking if you were to take the inoculating loop and dip it again and again and just do each sector with each dip? Or is it talking about dragging it from the last sector?
No, you're taking it for sector 1. You're going into the stock culture, picking up the cells, and then streaking it on sector 1. You then sterilize the loop because it tells you you're not doing that. And you go to the stock culture, and then you streak those cells on sector 2. And then you sterilize the loop, and you go to the stock culture, pick up more cells, and you streak those on sector 3. So that's
Okay.
how that one's going.
Okay, thank you.
Okay. Let me stop recording. Okay, there's a question about what chapters do the quizzes cover? And you can take a look at the syllabus. It will always have that for the next quiz, which is quiz 2, which must be taken this Wednesday, which I guess is tomorrow. It's going to cover labs 3 and 4, chapter 3, and the end of chapter 2. Okay? So
Okay, thank you.
look up in the syllabus.